But first, thanks so much for the invitation, opportunity to talk to you today about some of our work and as much as anything else, some of the ideas that we've had over the years about systems biology and subsystems biology. And I mean, gee, I think there's a lot of systems in this talk. Is there a lot of bioinformatics in this talk? Maybe not. Is there a lot of statistics in this talk? Maybe not very much at all. But hopefully you'll enjoy the story and join me on this journey, because you see the stuff I'm talking to you about today is a bit of a journey. I'm warning you now. So look, here's the dream. Here is the dream. So on the left, you've got a cell in parts or in pieces. On the right, you've got a cell. And I think, as systems biologists, what we want to have is a complete understanding of that machine as a system. A cell's a machine, isn't it? Basically, it's just a living machine. And of course, when we study cells, we do like to use omics scale methods and tools because you can measure a lot of stuff. You can measure the transcripts, you can define the genome, you can look at the proteins, you can look at modifications, you can try and measure interactions. There's a lot of stuff you can do with high throughput methods. And I think one of the things I really like about systems, and I've got it as a bit of a kind of a thought always in my mind, is that if we can understand a system, then emergent properties should come out of that. And you're probably wondering what's an emergent property. An emergent property of a car, for example, is that if you were to study all the individual pieces, you might get a bit of a feeling for, that this connects with that and whatever. But you may not understand that that thing moves. The movement is an emergent property. It creates heat. That's an emergent property. Heat is a property that cannot be broken down. Movement cannot be broken down. There's emergent properties that come from systems that are properties of the system itself. And it's very hard to understand some of those by looking at the parts. So systems biology for me is very much about, can you build something in a way where you can then try and understand emergent properties of that? But this is the dream. And look, it's hard because technology is not perfect. This is a small kind of car. It's still got a lot of parts. It's complicated, isn't it? And it's nowhere near as complicated as a cell. And so then you start to kind of break it down in a few ways. This is our cell rather than our car at the moment. But you might try and understand networks. And on the left, we've got a part of a signaling network. You've got protein interaction networks, gene regulatory, biochemical networks and pathways. But once again, the frustrating thing, and it's getting better, but the frustrating thing is, of course, all of these are kind of incomplete, aren't they? I mean, a genome you could say is complete, but you can probably look at a transcriptome deeply enough to say it's complete. But these are much more about interactions between things, aren't they? And they're largely incomplete or wrong. So it's frustrating. We're trying to understand systems, but a lot of the stuff we've got is incomplete or wrong. And typically, these are not joined well to each other. So if you're trying to understand a system, well, it's about joining these things, but our technologies work with these silos, and then how do you join them to get a view of a system? And so, of course, the question is, what do you do about it? Do you keep on working on these like this, or do you try and look at it a bit differently? And I suppose the argument I'm going to just put today is that maybe there's something called subsystems biology. It's a pretty lame kind of term for it, but you'll get the idea in that, well, instead of kind of looking at this in pieces, you might want to look at that instead. And if you break it down to the bits on the left, it's way less complicated. But maybe you've got a decent chance of actually defining the bits, defining the interactions, defining what it makes, which in this case is an engine, defining the emergent properties of that, because that emergent property, as I mentioned before, is heat, torque or power, carbon dioxide, emergent property, global warming, it's part of that problem, emergent property, can't break it down. And so the things you can then ask are, what are the parts, the interactions, can you define them? In biology, you need to think about what can you study in this way? Is there something which is sufficiently small, but not too small, that you can look at and maybe study in this way? And what's tractable? Can the subsystem that you're looking at be interpreted? Because we like to do things that you can interpret, because otherwise it's hard and you don't learn stuff. So complexity reduction and subsystems is a part of what I think I've been thinking about, what my group's been working on for quite a long time. So I will tell you a couple of stories today. They're connected in a way that you'll see as we kind of migrate from kind of story one to story two. And as I was chatting with Ellis and Elijah over lunch, some of these have been going on for a while, but there's good news in that the first one has reached a conclusion. It took us 12 years. You're thinking, geez, that's a long time. But hey, I mean, you'll see why I'm kind of interested and excited in it. So we're going to talk about defining a complete post-translational modification network in a cell, and then talking about how to connect a chromatin methylation system to the sensing signaling system in the cell. So let's start with this one. So many of you may or may not know much about post-translational modifications. I thought I'd drag out a slide from my second year lecture just to give you a bit of a reminder in case you're not in this space. A series of modifications are made to proteins after they're made. They're called post-translational modifications. There's a stack of ones which are common, and you would have come across phosphorylation, I imagine, being in the kind of environment that you are here. Acetylation, you may have come across. Methylation, maybe a bit less so. Glycosylation, fatty acids. I mean, there's a whole stack of these things. There's more than 300. The ones I've put up here are the most common ones that you'll find. They are relatively easy to detect. Mass spectrometry can help you a lot in terms of detecting these at scale. And I suppose if we're going to try and do this thing of finding all instances of a modification inside a eukaryotic cell, which is part of what we want to do here, the question is, can you do it? Can you find all sites of a modification, and how would you do it? And here I'm just saying, if you want to do that, you need to use proteomic techniques. Mass spectrometry typically, but things that tee up with that as well. But then if you're building a network, then the type of network we're going to build here is one where we try and find all of the enzymes that are responsible for all methylation sites. Because when you've got a modification on a protein, there's another protein that puts it there. Okay, it just doesn't turn up during translation. It gets put on after translation by another protein. So kinases put phosphorylation groups onto proteins. Methyltransferases that will become familiar with put methyl groups onto other proteins. And I guess, if we're trying to build this network and make it complete, we want to find all instances of a modification. We want to find all enzymes that are responsible for that. And then you start to build out a network because you've got some interactions between things that are happening. And with this, as we'll talk about, the genome, the literature, and enzyme discovery tools become your friend. But then there's this kind of other question. Well, if you are saying you've got all instances of a modification, you're saying you found all enzymes, well, how do you know that it's complete? Because, I mean, modifications are hard to find. I mean, there's a lot of proteins in the proteome. They've got a lot of amino acids on them. How do you know you found all instances? And how can you then make some bold claim to say this is complete or near complete? So we'll talk about that, and then there's that question that we've been exploring already about completeness reveal emergent properties, because that's what we want to find, properties of a system that you wouldn't otherwise be able to discern. Okay, so for those of you who might have come across any of the wet lab stuff that I've done over the last 15 years, you'll know that I'm a bit kind of obsessed with one of the smallest possible modifications you can make to a protein. Okay, so a lot of effort going into a small modification. It's where you put a CH3 group, a methyl group, onto a protein. Okay, so the joke often is, I mean, how much time should you spend on looking at this tiny modification? Why bother? But we'll get into that, I'll show you. Methyl groups you find them largely on lysines or arginines, but they exist on other proteins in the proteome, and they're on many high abundance proteins. And by that, I mean things like, ribosomes, you've got a whole stack of these things. Histones, you've got a lot of histones in your body. Every, 140 bases of DNA is wrapped around an octamer of histones. And you've got methyl groups on those histones. So there's a lot of methylation that goes on inside a eukaryotic cell. If we're to look a tiny bit more just at lysine, I just want to give you a small flavour of some of the stuff that this type of thing does. I won't go through its role in different amino acids, but just to look at lysine. So here you've got a protein without a methylation. You've got a methyltransferase, lysine methyltransferase. It puts on one methyl group. You can have that same lysine having 2 or 3 methyl groups. Unusual. Relatively few modifications are like this. You've got demethylases that can take it off. Okay, so you can put it on, you can put it off. You can have it in 3 states or 4 states. You've got a 0 state as well. You can ramp it up and kind of dial it back. Look, it's expensive. It costs the cell 12 ATPs to do one of these things. Whereas how many ATPs does it cost to do a phosphorylation event? Well, it's 1. Okay, so 12 times more expensive than a phosphorylation event. Lysine methyltransferases will show this, a very, very specific one or few substrate proteins. And in terms of physicochemical nature of this, you end up with the charge being maintained, but you do tend to have less hydrogen bonding that can occur between the side chain because you've got these hydrophobic groups here that are a feature of this modification. Some of the role, I won't go into this in detail, certainly very important in terms of the modulation of interactions of proteins. It gets recognised by other domains, can be important in the movement between the nucleus and the cytoplasm. There's a whole lot of stuff that happens with respect to transcription factors and, of course, histones. And you can find it kind of in competition with other modifications on proteins. But certainly a very important part of this is histones. And this is just a bit of a general view on a nucleosome. A nucleosome is just 8 histones with DNA wrapped around it. That's how a eukaryotic cell packages its nucleic acids. And the histones do have these tails. And what you see along any of these R2, K9, S14s, these are amino acids which are known to carry modifications. And you see that methylation's a feature. Okay, so anything which is a me on it is a methylation site. These happen to be the ones in the human cell. This image is from Brian Strahl's lab, who's one of the discoverers of this. And yeast has 6 of these methyl events on histone 3 and on histone 4. So you see this is important as we get a bit further into the talk. So what I will tell you is about this paper, and as we were discussing over lunch, I first had a grant on some of this methylation stuff in 2010. And look, it was one of these grants that, I mean, look, you're always proud of all your grants, but I mean, I was pleased with this one because I said, in this grant, we're going to find all the methylation events inside a cell. We're going to find all of the enzymes that act on that. We're going to functionally try and understand what it does. Of course, it was crazy ambitious, which is fine. ARC sometimes likes ambition. But then the thing which has been killing me for 13 years is, can we actually address this kind of aim that we set out to do? Because I wrote the grant in 2009, of course. And so this is it. So I'll walk you through this particular work. So, of course, what you do have to do if you're trying to find all instances of a modification in the proteome is you have to look pretty hard. Okay, so yeast has got 6,000 proteins. The average length of those is, what would you call it, 200 to 300 amino acids. So that's a lot of proteins, a lot of amino acids you have to look at very carefully to try and find these modifications. Now, for any of you who've worked in proteomics or with proteomics researchers, you'll know that proteomics is horrible. It's really horrible. Yeah, you've got this abundance range for proteins, which is just impossible to measure the low abundance stuff in. You've got peptides and parts of proteins that are very recalcitrant and not happy to be analysed. And looking for modifications, which can be low in the amount that they're on a protein, is hard amongst this other set of kind of issues. So the missing data kind of thing is just a feature of proteomics. So how can you find this type of stuff? How can you find all instances of a modification? So at least in our hands, the answer was not to do mass spectrometry and call it a day after a week of work. So the stuff that we worked through was a range of things. Certainly mass spectrometry is a key part of it, and that's our final kind of decider. But we used a whole stack of approaches over many years. You start off with your yeast. In some cases, of course, you can just push that straight through to mass spectrometry. Other cases, you might do gels. You might use some Western blotting, and here the bands that are being shown are those where we've probed with an antibody, so you kind of know where you might want to pick out the jellies you want to analyse it with mass spec. You can take proteins from the yeast, you can isolate the proteins or the peptides with antibodies that are specific for methylation, and then push that through to mass spectrometry, or in one particularly kind of exotic kind of way, we worked with a guy called Marc Snyder at Stanford. And he'd make these microarrays where he'd purified and printed down 4,228 proteins onto a glass slide. And he'd use that for doing a whole range of kinase assays. And we probed them with antibodies. And if you look really, really closely, if your eyesight is good, you might see some white dots. Yeah, there's some white dots. Here's some white dots, here's some white dots. I've got my glasses on, which is good. Here's white dots, white dots. They're in twos as well, aren't they? Whenever there's a white dot, there's a pair. And that's when we've probed with an anti-methylation antibody and then done this immunochemistry for the detection of it. And of course, after that, you then have to kind of find the protein somehow and do the mass spectrometry to figure out where the modification is. So there's a range of things that you can do to try and find methylation sites, but it's horrible because methylation, if you then start to look at the different studies that have reported this type of stuff in yeast, I guess the good news is that there's not hundreds of these. No one's found hundreds of sites in yeast. But then if you start to kind of, look at these a bit more closely, and we'll do that in a second, you'll see that there's some agreement between these studies, but there's a lot of disagreement. And in about 2015, 2016, Jean Hart-Smith in my group, fantastic postdoc, looked at this stuff really hard and came to this awful conclusion that large-scale mass spectrometry-based identification of methylation is subject to high false discovery rate. Up to 80% of the stuff which was described in the literature, at least in our take of it, was wrong. There's reasons for this. If you've got a single amino acid substitution in a protein compared to that in a database, if you're changing your glycine to an alanine, which is not uncommon, you're changing the side chain by 14 mass units, which is the same mass as an addition of a methyl group. If you are doing some protein prep where your proteins are coming anywhere near methanol and acid, then you will methylate proteins before they even see a mass spectrometer. And gee, people like to stain their gels, don't they? And use acetic acid and methanol. Yep. So you can see how this is a problem. And then there's other problems in terms of database searching and FDR discovery that won't go into just now. But I guess on the back of this study, what we did certainly kind of decide and use is what's called heavy methyl and heavy methylation. You can feed cells heavy methyl groups. There's something called S-adenosylmethionine. You have a heavy version of that, which then has a mass of 17 Daltons rather than 14 Daltons. And 17 Daltons is good. It's very characteristic. And then what you can do in your mass spectrometer, and those of you who've done proteomics might kind of get a bit of a feeling for this, you can, when you're finding what looks like a methyl group on a peptide, you'll have a light version of the peptide that'll turn up in your mass spectrum. And next door to it, you'll have a heavy version. And that's good evidence then that you've actually got a methyl group, which has come through biochemically and been put onto the protein in the cell. And there's software that we described here in association with Vincent Gogan's group, where you can then not just look at the peak difference, but also kind of look at how you have this correlation of these peaks throughout the elution of the peptide during the retention, chromatography of the peptides in mass spectrometry. So I suppose then you ask, well, okay, can you use all of this data in some way? Can you get this together? Can you get some view on what is likely to be the total set of methylation that exists in a proteome? And part of the reason I think why I like this and I like to talk about this is because that, at least in our understanding of the literature, this may be the first instance where someone has been brave or silly enough to say, well, yes, these are all the instances of a post-translational modification that have ever been described. Because you can't say what all the phosphorylation sites are. There's no way. No way. Can you say what all the acetylation sites are? Well, it's difficult because you get this enzymatic or non-enzymatic acetylation that occurs on proteins. So this is kind of interesting and kind of unique because I think it is the first instance where you can say, well, yes, we have found what we think are all the instances, or close to it, of a modification. But what's made it possible for us to say this is that we don't want to just say we've seen a modification a range of times. We want to find the enzyme that's responsible for it and putting it there. Because if you can show that you've got the enzyme that's put it there and you've got the site, and you can actually independently show that in some other assay that that's what happens, then you're pretty close because you've got the evidence that's there, you've got the enzyme that's put it there. And so can you then do methyltransferase discovery? Can you find those enzymes which have actually put the methyl site onto that protein? Now, the great news is that methyltransferases have a binding domain for S-adenosyl methionine. That's the methyl donor. In the same way that kinases have an ATP binding site. And in yeast, there's these numbers. So there's that's about kind of, 72 enzymes like that. But of course, they just don't methylate proteins. They can methylate DNA, they can methylate RNA, they can methylate fatty acids, they can methylate small molecules. So you need to find which ones of those are actually methylating the proteins. And there's a lot of work that was done by Steve Clark's lab in this area, a lot of work that was done by Paul Fallner's lab in this area. There's other literature, kind of things that were going on. And we did get involved in a pretty dreadful race with Paul Fallner's lab and Steve Clark's lab because we're all onto this. We'd narrowed down the set of methyltransferases that must be doing this methylation. And then there was this race that we got caught up in between those three groups about all this stuff. So certainly, I mean, the part of the idea is that if you've got a methyltransferase that methylates a protein, if you knock it out, you might see something like this. Okay, so this band is where you've got methylation. You knock out, in this case, ribosome lysine methyltransferase 1, and you see a loss of that. So loss of methylation when you knock out. Of course, what you need to do is not just showing that you're losing something. You do mass spectrometry on this to find the exact site that you've got or lost. You need to then have a gain of methylation assay where you recombinantly make the protein, the new enzyme. You purify or have available the substrate protein. You do the in vitro assay, and you do the mass spectrometry to show that you've got the same thing. So you have this gain and loss and gain of stuff going on. And look, we had a lot of fun with this stuff. Here's some of the papers that describe some of the new enzymes which we have discovered. There's another two, which I'm not showing you here, which are human ones, because we had a bit of fun doing the human ones of these as well. But I like these because these are new enzymes. Before that, they were known to be methyltransferases, but their substrate protein was not known. The exact position where they methylated was not known. So this is old-fashioned biochemistry with a bit of a twist. New enzyme doing protein methylation of, in most cases, one amino acid in the cell. So wave the flag for biochemistry. It is still possible to discover new enzymes if you want to do so. And it's nice because you discover them and then they're yours. I mean, you found them. They're kind of like your children. They're lovely. So what we then did is we said, well, okay, we've done a lot of enzyme discovery, and we got ourselves to the point where we think there were no more enzymes to be found that were protein methyltransferases. That was super important because the other ones that were on the list, clearly they were RNA ones or they were DNA methyltransferases, but we very firmly felt that all of the protein methyltransferases had been found. So then if you cross-match those against the methylation sites that have been described in these 9 or 10 papers, you should be able to see, and best of all, the methylation sites that have consensus are green in that they match to a known methyltransferase. So here we're cross-matching all of the sites that were discovered with the known methyltransferases. And at the same time, there was a range of sites in blue which we politely called may be associated with unknown methyltransferases, but much more likely is these sites are just wrong. And I'm happy to own some of those sites that are wrong. You'll see some of our work there is we've described sites which we think are wrong. That was our earlier work, so we'll put it at least down to that. But here you're seeing certainly really good kind of matching of the conserved sites between all these studies with the known methyltransferases. And certainly in and amongst this, there were consensus sites with methyltransferases. Few methyltransferases have no known site. The stack of stuff which is wrong, and the closer we look at that type of stuff, we think it is just, it's error, false discovery. These ones at the end here, you're probably a bit worried about those. They are ones that all map to the same methyltransferase, which is a very particular type of methyltransferase, has many proteins that it puts methyl groups onto. Whereas most of these ones through here, they are a single site of methylation that is put on by a single methyltransferase on a single protein. This one-to-one matching and mapping, really, really interesting. So we've got this, this network, near complete protein methylation network in yeast, so landmark for a modification in a proteome. You'll see that the things in blue are the methyltransferases, the things that are red are the proteins they act on. You'll see that there's a lot of one-to-one mapping, and when you've got this one-to-one mapping, in almost all cases, it's one site, one amino acid. Exquisite specificity. You'll see that the proteins are and this is an emergent property. There's a lot of proteins in translation that are methylated. A lot of proteins in RNA processing that are arginine methylated. The histones are lysine methylated. And then you've got a few others here. And you see there's some that have these kind of, slightly kind of different colours and borders to them. These are ones that we're predicting, at least the methyltransferases, in some cases the substrates. And look, gee, if anyone wants to join my lab and kind of tidy up these ones, you'd be very, very welcome. That would be a treat. Chromatin, I've just highlighted that because we'll talk about that in the next part of the talk. So I guess insights here. So we've methylated proteins, they're in important functionally related groups. Most of the methylated proteins are essential. It's an emergent property. They're important proteins that get methylated. Many are sequence constrained. But what we thought was really interesting is the enzymes that put it there are non-essential. And that's kind of funky, isn't it? You think that, well, important proteins, methylation on important proteins should be kind of essential, but they're not. So then you've got this question about, well, why are they there in the first place? Certainly they don't show redundancy like you see in signalling. Doesn't have that at all. So, I mean, working hypothesis means to fine-tune function. Tiny improvements in key processes. You add a methyl group, you change the structure just a tiny bit. Provides evolutionary advantage. And is there evidence for this? Well, I think this is super strong. 5 methyltransferases act on elongation factor 1 alpha. For those of you who love your ribosomes and the biology of that, elongation factor 1 alpha brings tRNAs to the ribosome. This is the protein inside the eukaryotic cell that has the most attention for methyltransferases. There's 5 of these that have evolved to act on this protein, and they are independently evolved. They're not of the same lineage. So nature has done this thing 5 times because this is one of the most important proteins in the cell. It just keeps on feeding the ribosomes with tRNAs. If you can do that a tiny bit better, a tiny bit more efficiently, a tiny bit more accurately, you will win in evolutionary time. Because to grow, you need proteins. If you can make proteins better than the other guy, you'll win. So really kind of interesting set of observations here. And this is a system, isn't it? We've defined a system. These are some of the emergent properties that we can start to see from that. So let's kind of jump into our second story. And this is connected to our first one because of you'll see it in just a second. So we're back to methylation. You see this is a bit of a thing for me. It's been going on for a while. I apologise. How can someone get so enthusiastic about it, CH3 group? But you'll see that in yeast, so we've got our histones. This is our, we have 4 of them here that forms the octamer, two of these come together. In yeast, you've got histone 4, it's got 3 methylation sites. Histone 3 is where there's a bit more activity, and you've got these sites, plus you've got demethylases, these one in pink that can remove it. And nice thing about this whole thing is that you've got 4 enzymes that can put methylation onto the histones and 4 that can take it off. It's a beautiful subsystem in its own right straight away, isn't it? And then when you start to look at I guess, how histone methylation exists, you can look along the genome, can't you? And you can look at how you've wrapped up your DNA around your nucleosomes. And you can look at the methylation that exists along the genome. You can do this with kind of chip-seq type approaches. But the thing which is just incredible is that if you look, for example, at where you've got lysine 4 methylation on histone 3, so H3K4, you see, if you've got a gene, the promoter may not have very much of the monomethyl, but then you see it in the coding region. The tri is kind of the opposite. You see trimethylation at histone 3 lysine 4 at the promoter region, but not much down there. And then for, if you look at histone 3 lysine 36, you see methylation, you've got mono at the start and then di and tri. And then for something like dot 1, which is methylation on lysine 79, it really is just across the coding region of the gene. This is exquisite. The enzymes have to put them on there, don't they, in some way? They have to know where to go. They need to know where the genes are. They need to know where the intergenic regions are. They need to know what the start and the end of the gene is. And so you've got these 8 enzymes, those are putting it on, putting it off, that have this type of stuff, these crazy patterns along a gene. And it does this for every gene. And the whole thing's dynamic. It's changing all the time as the cell is adapting to whatever is happening inside itself or the external environment. This is exquisite. And the question is, of course, well, how is this regulated? Or who's controlling the controllers is the name of the grant that we wrote. Everyone likes a catchy title. Who's controlling the controllers? And so this is when we kind of start to think of this as a bit of a model here. So we've got our external or internal signal. This is the kinase, a part of the sensing system of the cell. And I guess we're thinking, well, maybe the demethylases or methyltransferases are getting phosphorylated, and that might then make them change the methylation on the histones and the nucleosomes. But I think the thing which I like particularly about this is also that what we're looking at here is really the intersection between 2 subsystems of a cell. One is the sensing and signaling system, and the other one is the kind of the chromatin. And chromatin, of course, affects gene regulation and gene expression, doesn't it? And right in the middle, and isn't this gorgeous, are these 8 enzymes. There's no other way that the methylation gets put on or taken on. This is it. You've got 8 enzymes that are doing this. The human cell is horrible, of course, because you've got about 80 which are doing all of this, but this is only 8. So is it tractable? Well, yeah, kind of. Will we be able to look at this as a system? Well, yes, maybe kind of. You can see how we've got some subsystems biology stuff going on here, which should be something we can learn stuff from. And look at this. I mean, this is what I love about it, because information has to come from up here to get aggregated on these enzymes, these 8 enzymes, before they then go and do stuff down here. They are that essential kind of pinch point between the sensing signaling system and the chromatin modifying system in the cell. Should be a great thing to study. And it is. And so we can make some predictions. So of these enzymes here, so we're kind of saying, well, maybe phosphorylation will be specific to cell state or stress. Some phosphorylation events should happen just in certain situations. Stands to reason. The phosphosites, if we change them here, should change the methylation, which is happening downstream. The loss of a phosphosite, if we lose this site, should affect the cell's response, because information cannot get from here down into here. We've removed that communication point. And of course, with a bit of luck, kinases, the things that are upstream of here should be discoverable. The great news being that inside yeast, there are 120 kinases, which is not too bad. compared to the human cell with about 470. Okay, so we're in the bounds of tractability here. So if there's a stress event, well, you might think that a kinase might then, say, phosphorylate this and then have these downstream consequences. So you can kind of see where I'm going with this in terms of how we're going to study this. So the question that we asked is, are those histone methylation enzymes phosphorylated? And to do this, we purified each of the proteins, the 8 proteins. We did a whole stack of mass spectrometry. We cut them up in 4 different ways. We used 2 different mass spectrometry techniques. You can see the coverage that we're getting here. Black means we found it 8 times. As you go up and down the scale, you found it less times. And this is pretty good for a big protein of 730 amino acids. It's a big one. But we did this for all 8, and this is what you find. So we found 143 phosphorylation sites on the 8 enzymes. There will be more. I'm not saying this is complete because we only looked at them under one condition. Okay, but it's a decent start. 143 sites. Some of them are clustered, and there's some funky stuff going on in that you see that in the methyltransferases, those that put it on, you can see there's methylation that are in the active parts of these enzymes. These ones over here, these are funky because these ones do not have any phosphorylation sites in the actual domain, the part of the enzyme that takes stuff off, but it has it completely in the disordered regions. So there's funky stuff going on here. I won't go into too much of that. It's all in the paper. So we got these sites. And then, of course, if we zoom in and look at just this one here for a moment, this is one of our demethylases. You can ask that question, well, if we subject the cell to a stress, do some of those phosphorylation sites change? And this is part of our hypothesis, and information should come from the sensing signaling system. It says, okay, there's salt stress, and it should feed down into this enzyme and some others and change the phosphorylation. And if you do quantitative mass spectrometry of sites, then you start to see some of this, and sorry for all of the bars and whatever going. Look, some statistics. Yep, not too much today, sorry. Hopefully I did the right test. But what you do see here, which is fantastic, is when you treat the cell with 1 molar salt, and you then analyse the level of phosphorylation at these different phosphorylation sites. This is just S412 means serine 412. You see that there's an increase in phosphorylation on treatment with salt. So this means information is coming from our sensing signaling system into our histone modifying enzymes, which is good. And there's a stack of these, okay? Some don't change, but some do. And look particularly at, for example, S412. It's a really nice change. What we can then do is then say, well, let's start to mutagenize these. And if you do this, in this particular case, Nicola did a range of mutagenesis where she generated 10 different mutants. This is all by genome editing by homologous recombination. And she did these in groups in this first instance because it's just a bit more convenient to do it in groups. These things are next to each other and saving Nicola from having to do 40 of these things. Just did 10 instead to start. Because the prediction we're making is if we mutagenize the site, then information is no longer getting through from sensing signaling down to chromatin, and the growth of the cell should change. And so if we then do that experiment, so these mutants show slow growth in salt. So if you mutagenize S412, which is this one here, the big change, what you then start to see is that in increasing amounts of salt, this mutant which contains S412 starts to grow more slowly. It cannot adapt to salt. And if you get a bit more granular, Nicola's an amazing student. Nicola then went and built the single site mutants for these. And once again, for serine 412, again, if you mutagenize it and change it to an alanine, it can't be phosphorylated. 5 minutes? Perfect. Then what you see is the cell can no longer grow under that type of stress as effectively as the wild type. So the information is not getting through. If you look at these salt-responsive sites, one of the amazing things is you start to see patterns. They're in SP motifs, and those, for example, at S412 is particularly an SP motif. And so the question is, can you find the kinase? Can you connect this to what's upstream? 120 kinases in yeast, but only 6 are known to be SP kinases. Okay, so there's the motif. One of them known to be associated with osmolarity and osmotic stress, HOG1. And so can you then ask the question, is HOG1 the kinase that puts this phosphorylation on? And just to summarize, enormous amount of work. If you do an experiment where you've purified recombinant HOG1 and you've activated it with another protein, which is the kinase itself that activates it, you have to express and purify that. And do an in vitro experiment. Can you see an increase in phosphorylation on the RPH1 protein? The answer is yes. You get this phosphorylation occurring when you have activated kinase mixed in with your protein. This is our negative control. We've knocked out the active site of this one just as a good negative control. And if you do this for the whole protein, you see a certain number of sites where you get dramatic increase in phosphorylation by the HOG1 kinase of this RPH1 protein. Now, there's a stack of other stuff going on in here. I won't have time to talk to you about it, but you're probably wondering, why is this phosphorylated like that? Happy to answer questions. So RPH1 phosphorylation. So what we did find is some salts were found to increase on, some sites were found to increase on salt stress. If you mutagenize these, the cell can no longer grow as well, presumably because it's no longer getting that change to its chromatin which it wants and needs. Many of these sites here, or on SP motifs, and the HOG1 kinase was shown to phosphorylate that. So we're saying that we've now been able to work backwards, and in doing so, we have got that connection between the sensing signaling system and the chromatin system of the cell. It's not all roses, or beer and skittles, as some people say. Look, here's work that Ryan did, 30 genomic mutants for DOT1, did a whole lot of stress tests, found nothing. But the good news is, and I might just skip to the second one of this example, another demethylase, we only found it to have one phosphorylation site. It was easy to pick which one to work on. And if you mutagenize this site and do stress screening, we found that all of a sudden, cells that have this phosphorylation site mutagenized to alanine cannot grow effectively in the cold. So that single site is where information comes in. It's actually on the PHD domain, the DNA binding domain of this, and it can no longer grow in the cold. Now, it doesn't affect the histone methylation. There's a whole lot of stuff going on here where histone methylation was affected, but certainly you see this very strong phenotype. So these things are important in terms of stress response and then how a cell can grow. Okay, so I think we've been through this. We found phosphosites associated with stresses. Phosphosites can change histone methylation. Loss of the sites can affect phenotype, and kinases are discoverable, linking the signaling to the chromatin system. But just more broadly, I mean, subsystems biology, so our protein methylation system, we have our sites of methylation, our methyltransferase mapping, we think it's near complete. Function in most sites not understood. And this is the problem with this type of stuff. I mean, functionally analyze these things is hard. We've done a lot of functional stuff, and we found some effects, but it's hard. We think it probably is a means to fine-tune function. And chromatin methylation signaling, so the methyltransferases are regulated by phosphorylation. We've got some of our first links between the sensing signaling system to chromatin. Some sites are of clear purpose. And look, this is the challenge for us. Now we've got kinases. We've got 153 phosphorylation sites on 8 histones. Oh, sorry, histone methylating enzymes, 6 methylation sites on histones. And so can we find the function associated with all the sites? Can we find the kinases And so invite me back in another 15 years. And I'll give you the talk where hopefully that subsystem will then start to be complete. Okay, I think I've come in 1 minute under your 5 minutes. So acknowledgements, here's the current team. They're all fantastic. Some of the people who have done very large amounts of the work today are Josh Haney, Jean Hart-Smith, Ryan Saprovich, Nicola Karakatsilis, Tim Kudis, Jason Lowe, and Aidan Tay. And thanks very much again for the invite and for your attention.